Hi, welcome to my video. Today I'll be talking about fungal DNA extractions and PCRs. What is DNA extractions and PCRs? Why is it important? DNA extraction is when you select a specific segment of DNA and amplify it using different extraction methods. Then we can sequence it to obtain genetic code, which can be used for a variety of things. Some of these things are identification of species, understanding how endangered a species is, and differences below and above ground in terms of fungi. So how do we do this? Well, step 1, attaining a dry fungal sample. There are two parts to a mushroom, the fruiting body, which is the top part, and the fungi, which is underground. Step 2, choosing a DNA extraction method. First we have CTAB and alkaline. CTAB is very lengthy, while alkaline is very short, but they were both made as cheap alternatives to more expensive DNA extraction kits. Now that we have our dry sample, we can start. I'm going to explain the alkaline method because it's a lot shorter as well as being used specifically for fungal DNA. Two things have to be prepared for the alkaline method beforehand. Extraction solution, which breaks down cell wall, and a dilution solution, also known as the BSA. This is the alkaline method completely. I'm not going to go through it all because it's very long, but once you finish, yippee! You now have a sample ready for PCR. Step number 3, running a gel. First, you have to make a gel. To make a gel, you'll need agarose power, TAE buffer, and ethidium bromide. Now that you have your concoction, you can pour it into the mold. This mold has specific little cones that will allow you to put in your samples later. While our gel is drying, we can prepare samples. To do this, we first have to mix in our sample with some blue dye. This will help it be more clear and visible in the PCR. Now that our gel is dry, we can pipette some of that sample we just prepared into each of the notches. Then we can put it into this machine, which consists of a chamber and buffer, which helps run 200 volts of electricity through the gel. Time for step 4, results. After we take our gel out of the chamber, we can run it under a UV light to see if it worked. It will look something like this, where some of them did amplify while others did not. Well, what if sometimes it doesn't work? That's okay, you can modify the alkaline or the CTAB method to see if you get more results. With the ones that did work, we can put them into a DNA analyzer, which is a very expensive piece of equipment used to sequence DNA into their base pairs. Now that we have the base pairs or the DNA sequence, we can do a variety of things with them. For one, we can go into the National Library of Medicine's GenBank and compare our results, or build phylogenetic trees, or for archival purposes, as well as a lot of other ones. Thank you for watching, and thank you to the Chicago Botanic Garden, Field Museum, and NSF for funding this opportunity.